to the National Association of Plant Breeders webinar series, How to Breed New Plant Varieties, Imagining and Engineering Crops. My name is Shelby Ellison, and I'll be serving as your host for the webinar. Today's special presentation will be given by Professor Jim Myers from the Department of Horticulture at Oregon State University. Dr. Myers' research group aims to increase the availability of organically produced vegetable seed by identifying and breeding varieties adapted to organic systems. Today, Dr. Myers will be speaking about pea breeding in his presentation, Putting the Snap in Peas. Putting the snap back in peas. Now I'm going to switch control over to Dr. Myers.
Thank you, Shelby. Before I get started, I just wanted to acknowledge the help of Abby Graham, a graduate student in my program, who helped me put this presentation together. So, that's okay. Here, first part that we're going to be talking about is some of the characteristics of peas in general, and then we'll get more specifically into edible potted peas. The pea is Pisum sativum. It's a cool season legume. It's frost hardy when it's in the vegetative stage, becomes less so once it's gone into reproductive phase. It's a diploid with 7 pairs of chromosomes, fairly easy to breed because of that. It's also self-pollinated, and the pollination is self-pollination is enforced by the anthers dehissing 24 hours before the flower even opens. You can see some pea flowers in this image here of the intact flowers, and then one that's been taken apart, so you can see the various parts to the flower here. You have the carpel surrounded by an anther sheath with the anthers attached to it, a style and stigma, and then that is surrounded by a pair of petals called the keel, and then you have 2 wing petals out to the side, and then the banner petal. Peas are generally propagated and sold as pure lines, as varieties. No one's ever really devised an economical way to produce F1 hybrids with peas. Peas are quite an ancient crop, domesticated somewhere between 8,000 and 10,000 years ago in the Fertile Crescent area, which is modern-day Jordan, Syria, Iraq, Turkey, in this particular area. It's also been very historically important played an important role in genetics, with being the crop that Mendel worked with to devise the laws of genetics. But even before Mendel worked with peas, there were other breeders clear back in the beginning of the 19th century that were systematically developing some of the original breeding techniques that were used in peas. Some of the changes that happened under domestication with pea was it's similar to what has happened in other legumes. There's been a reduction in seed dormancy, shift from pods that shatter very easily to pods that shatter less easily, an increase in seed size. Wild peas tend to be purple flowered and have colored seed, and there's been selection for white types out of this. There's also been a shift in cotyledon color. Wild peas tend to be yellow cotyledon color with a round shape, such as you see here. And there's been types derived which are green cotyledon, represented here on your right, also with the wrinkled seeded trait. Wrinkled seeded trait is something that's used almost exclusively in garden peas. Pea production happens in the U.S. In different areas depending on whether it's dry pea or the garden pea. Most of the commercial dry pea production is in the Palouse area of Idaho and Washington, or the Mindak region in North Dakota and Minnesota. Green shell processed production happens in many northern states, including New York, Wisconsin, Minnesota, Washington, Oregon, and Michigan. And a snap pea, there is some processing that is done in the Columbia Basin and then in the Magic Valley in Idaho. And then finally, there's fresh market production, primarily winter production, that takes place in California and Florida, as well as elsewhere in the U.S. On a worldwide basis, peas are grown, again, primarily in these more temperate, cool temperate zones. You can see quite a bit of production in Canada, through Europe, in China, in the highlands in India, in Australia, and in highland regions in Africa as well. So there's both dry and processed pea production being shown here. A lot of the snap pea production, particularly on a worldwide basis, is done for shipping into winter markets in Europe and the Americas. In Africa, we have production for the European market, and then Central America and South America is where you see production for the U.S. market. I've kind of backed into what the different types of peas are, but here's something a little more formal on describing these. The field or the dry grain peas, where the mature seed is eaten, and then the other class, a general class, are the vegetable peas. The vegetable peas include things like the shell peas, pictured here at the top, and which has basically it's just the seed inside that is eaten. But then there are two types in which the whole pod is eaten. These include the snow peas, or Chinese peas, sometimes they are called, and then the snap peas, shown here at the very bottom. And you can see that they have different shapes that are characteristic of each type. And in a little while, we'll get into some of the traits that make these different shapes and the reasons for why they are in peas. This chart here gives you a little more definition on the market classes. We have the different types here in the first column, and then for cotyledons, how they differ for the cotyledons, the pod, the plant, and then the parts that are consumed. If we look at field pea, these can be either yellow or green cotyledon color, and they tend to have smooth seed. There is pod fiber present, but the walls are thin. Plants are generally quite tall, and it's the mature dry seeds that are consumed. With garden pea, generally these are green cotyledon, mostly wrinkled seeded, but there are some smooth types. These are mainly the early Alaska types. There is pod fiber present, and the pod walls are thin. The plants are generally short. This is particularly true of processing types where mechanical harvest is used. And then it's the immature seeds that are used that are shelled out of these types for consumption. Snow pea has green cotyledon color generally, although you can find some yellow types. The seed are both either smooth or wrinkled. It really doesn't matter in a snow pea because you generally eat it when the pods are so young and the seeds are so young that the seed characteristics don't influence the flavor. In these types, there is no pod wall fiber, but the pod walls are thin. The plants can be either tall or short, and it is the very young pods that are part consumed. Snap peas generally have green cotyledons. Their seeds wrinkled. The parchment is absent, and they have thick pod walls. And they're mostly short. There are a few tall types, but particularly for processing, you want short again. And then it's the immature pods and seeds that are eaten, but they're more mature than what you find typically in a snow pea. Okay, so we're going to get into a little more detail on the traits. One thing I want you to notice here is this curvature in the snow pea that we have. The cupping, this is very typical of these types, and it figures into our story in a little bit. So peas have been around for quite a while, as we mentioned at the very beginning, but the historic information on the edible pot of peas is not as extensive. We do find things, for example, like this painting by Turkowski from about 1795 that shows snow peas in a still life here. Again, notice the cupping of the pods and the the, the lack of straightness in these types. So peas, the edible podded peas were most likely derived through stepwise mutations in existing field peas and garden peas. Basically, people would find mutations that eliminated fiber in the pod and would preserve these types, and then that would become a snow pea. Dwarf gray sugar, for example, you may be familiar with that variety. That's one that comes from a field pea background, whereas something like mammoth melting sugar probably comes out of a garden pea background. Snap peas arose also from snow peas with a second mutation that would change the pod wall thickness from thin, as you see here in this picture for the snow pea, to thick with the snap pea. Most of the modern day snap peas have white flowers and wrinkled green cotyledons, which suggests that they have garden pea origins. And many of the most recent ones have been actively bred and coming, interesting traits from things that were not snow peas or snap peas. But in the past, this is the kind of the stepwise pattern that these types have come about. There is historical evidence for snap peas being around quite some time ago. I've found reference in an herbal by John Ray from 1686 to sickle peas, which are probably snap peas. He talks about the pods being edible and they are very easily destroyed by birds and things like this. Vilmorin in his vegetable garden described what he called butter peas, which based on the pictures were snap peas in the description and pictures. Wellensink, a pea researcher in 1925, was working with what he called the great butter pea, which appears to have been a snap pea. Hedrick in 1928 in the classic Vegetables of New York in the pea book described a butter sugar group where he knew of 6 varieties that fit this group, and these were described as being edible podded, succulent, thick-walled, and that sort of thing. So they've been around, but they've always been just kind of a minor vegetable. And so why is this? Why haven't they been more popular in the past? A lot of this can be, I think, attributed to their popularity today can be contributed to the work of Calvin Lambourne, who was a pea breeder at Galveston Seed Company for many years. The first variety he developed was Sugar Snap and was released about 1979. What he was trying to do, he was given the problem of solving that pod distortion that I pointed out earlier in the snow peas. And he thought, well, what would happen if he crossed this rogue type that was found in a garden pea dark-skinned perfection, which was thick-walled. It was the N gene. And what if he crossed that to a snow pea, and he crossed it to mammoth melting sugar? And he discovered that, yes, it does straighten out the pod, but you get something totally new and different. And so this was the origin of the snap pea. And at that time, he wasn't aware of these types that people had worked with before. But he jumped into very enthusiastically promoting these types and pretty much single-handedly increased the popularity of these types and got them going. Most varieties that you grow today are going to be ones that came out of his program or have been derived from his material. There are a few that are not. For example, at Oregon State University, we have a variety called Cascadia, which comes out of our program. But for the most part, the snap peas out there are all from Calvin Lambourne's program. Okay, some of the traits that are specific to snap pea. Here is pod fiber, or the called parchment. You can see in the picture here the high fiber pods tend to be very smooth, and as they dry down, then they maintain their shape, whereas low fiber pods tend to become wrinkled and distorted. This middle pod is a snow pea, and this bottom pod is a snap pea, and you can see that there are differences in the shape that are specific to snap pea here. There are two genes that control the expression of pod fiber. When they're present in the dominant form, you get fiber entirely present. If you're recessive at the V locus, you get little patches of fiber distributed throughout the pod. If you're recessive at the P locus, you get fiber primarily along the dorsal and ventral sutures. There's a separate gene that controls the fiber in the strings or the pod sutures that we'll come to in just a moment. But when you put P and V together, then you pretty much have absence of fiber in the pod wall. Pod wall thickness is another gene that makes the snap pea unique. This is controlled by the N gene. It's again a recessive gene expressed in with the thick pod walls. It has pleiotropic effects of reducing overall pod size. It tends to give pods a round cross-section, and they tend to be more curved than with the dominant N. Basically what's happening is there's an increase in the number of parenchyma cells, and there's also some increase in cell size as well. Another gene that is important, you can have snap peas that are stringy or snap peas that are stringless. What I mean by that is that they have a string, they have or don't have a string in the pod suture. This is a stringy type here where the pod strings has partially been pulled, and then here's a stringless type where we did the same thing with it. Stringless this is controlled by the Sin2 gene, or Sin which means without strings in Latin. It's a temperature-dependent gene where under warmer conditions it's more strongly expressed, things are more stringless, and under cool temperatures you'll tend to see a string present in the pod. It also affects other plant parts as well. Pods and plants will be somewhat shorter, smaller. It becomes more pronounced under heat and in stressful environments. So stringless types can be, if you try to grow them in particularly warm conditions, will be very non-productive. And in fact, the seeds planted into warm soils will sometimes not fail to come up just because they're of their weakness that is enhanced by the warm conditions. The stringlessness is controlled by a single recessive gene, and it shows distorted segregation ratios. You get way fewer stringless types than expected if you make a cross and you look at the F2 generation. And what causes this is that the Sin2 gene also reduces pollen tube growth rates, so that the pollen is less competitive than normal pollen, and which causes the distortion in inheritance. Another effect of the Sin2 gene is that when you combine it with the wrinkled trait, seeds are dimpled rather than being wrinkled, and this is confusing sometimes when you're making selections out in the field. So now on to snap pea breeding here. Crossing, by the way, in snap peas is pretty easy. They're large flowers, easy to work with. A pair of forceps is about all you need to do that. Techniques for self-pollinated crop work very well for snap peas. They would include things like pedigree method, single seed descent, backcross breeding for introgression of simple traits, and then inbred backcross method for introgressing quantitative traits, recurrent selection, and bulk breeding. What I use in my program primarily is pedigree method. And this allows you to make selections in early generations for the important traits that you might otherwise carry along in a technique like single seed descent. So it's fairly basic in terms of the techniques that are applied. Some of the breeding constraints and advantages for peas in general is that they have a limited, this is a constraint, they have a limited seed multiplication ratio. For every seed you plant, you get approximately 10 back. So it can take several generations to really increase your crop. There's a low crossing efficiency. This is another constraint. Crosses are made by hand. It's time-consuming process. You get 2 or 3 seeds per cross. And this is the reason why people have never devised a way to make F1 hybrids in peas. And the heterosis is certainly there, but we've never had any good sterility system and crossing system that would facilitate F1 hybrids. Peas do have a high selfing efficiency. That's what they do. So that's it's very easy to carry them forward with selfing. They generally have short reproductive periods. They're early season, so you get them in the ground early. You're generally out of the field by sometime in August with your planting. So that you can go back in pretty quickly with a generation in the fall greenhouse and then another in the spring greenhouse. You can run about 3 generations per year in terms of generation advance. Peas have large seed, which can be an advantage or a disadvantage. You can see various traits with them, that serve as genetic markers. But on the other hand, the seed is heavy and bulky, so it takes a lot of space to store. It can be costly to ship in any quantity. So breeding specifically with snap peas, if you're working with, it's going to depend on the type of snap pea that you're working with. So if you're dealing primarily with stringy snap peas and you're crossing stringy snap by stringy snap, there really are no special considerations that need to be taken into account. You just apply your regular breeding techniques. However, if you're crossing it to garden peas or to field peas, then you're going to be segregating for many traits in the F2. And you have, say, 5 basic genes to get back to the stringy snap type. Only in an F2, only 1 in about 1,024 will have that phenotype, the complete snap pea phenotype. So you can improve the odds by making a backcross to your snap pea parent and then selecting out of the backcross. It gets more complicated when you're bringing with stringless snap peas. Stringless snap by stringless snap, you're working in the same background, so there are really no special considerations. But if you're trying to integress traits from stringy peas, whether they be stringy snaps or garden peas or field peas, you're going to get this low recovery rate of the stringless phenotype in the F2, and you either have to grow enormous populations of F2s to select. We're talking maybe 5 to 10% of the plants will be stringless in that material. Or the other technique is to use backcross, but the backcross has to be done in a particular way. I'm showing the female parent here first, and then the male parent in my crosses, and it doesn't necessarily in the first cross matter which is the male and which is the female. I have this with the stringy parent being the female and the stringless parent being the male, but it could be done the other way around. So you get a heterozygous F1, and then backcross that to your stringless parent, but make sure the stringless parent is the male. This is done to prevent pollen competition. You're putting homozygous stringless pollen on this heterozygous F1 female, and so the pollen, all the stringless pollen will fertilize, has an equal chance of fertilizing either the stringy allele or stringy type or the stringless. And then you'll get approximately a 1-to-1 ratio of heterozygous to stringless, and you throw away the heterozygotes and keep the stringless and move forward with your breeding program with those. Now, some of the other traits that are not necessarily unique to snap peas, but they are important in snap peas, I wanted to go into a little more detail about these. These would be seed shape, wrinkled or round or dimpled. This is controlled by the rugosis gene or the R gene. It's a recessive again. It's basically a defect in the starch branching enzyme that slows starch accumulation and increases sugar content in the seed. And eventually, even in a wrinkled type, you will get, in a mature wrinkled type, you'll get starch and it will begin to taste bitter. But there's a period of time where those types will taste very sweet. There are other loci for this particular trait. There's an RB gene that is sometimes used, and RA and RB have combined and produced a sweeter type, but there are other loci that are known, not as well known. They have potential too, and that's maybe something that someone might want to investigate someday, is the effect of these loci on flavor and other characteristics. This picture shows wrinkled, dimpled, and round. The dimpled here is what happens when you combine the R locus with the Sin locus. You get, it changes the wrinkle to a more of a dimpled phenotype. Cotyledon color is determined by the I gene. Yellow is dominant to green. You can see the cotyledon color here in dry peas. And most snap peas are green cotyledon. Pod apex shape is something that is important in snap peas. The BT gene controls the shape where the dominant allele produces blunt pod apices, whereas the recessive allele produces the pointed apices, such as you see here on the right. The BT has a couple of effects in snap peas. One, it helps with shedding the senesced blossoms off the tip of the pod. And this is important because if it's retained, then thrips will get under there and scarify the tissue. You may get pathogens that get a little bit of a foothold, the saprophytes that'll get going there. And so it's important to shed that, because in snap peas, any sort of pod blemish makes it unmarketable. So the other thing about this gene is that BT pods tend to be straighter, whereas the acute apex type pods will have a bit more of a curve, which is less desirable. Anthocyanin pigment is important in peas. The A gene produces purple flowers, and you can see various examples here, kind of the wild type here down at the lower left. Here's a variation in the anthocyanin pathway that produces more of a reddish hue. Here's one type that produces kind of a greenish flower color, that probably a lesion at the beginning of the anthocyanin pathway. And then here's the recessive A gene that produces completely white flowers. You also get protection of color in other plant parts. It can occur in the stems, the pods. You get purple flecking on the seeds, and there's a colored ring in the maculum, which is red stipules join the stem of the plant. One of the things about having anthocyanin present is that it causes pods to be more astringent. This can influence the flavor of edible potted peas, this type of astringency. Let's see, I'm not able to advance here. Okay, plant height is another gene. This is controlled by the LE gene, with tall plants being dominant to recessive. One thing of importance to breeding snap peas is that LE is linked to one of the pod fiber genes on linkage group 3. So depending on the cross you're making, you may need to break this linkage. In the pictures here, we have tall plants, and when we have short plants, there's another trait that these short plants have, which is the aphylla leaf type, which, when we get to the next slide here, well, the aphylla leaf type causes the leaflets to be replaced by tendrils in the on the leaf. These types have found a place in commercial production where the aphylla types will have reduced lodging. They grab together and they grow much better under those conditions. Interestingly, aphylla types tend to do better under heat stress, perhaps because they have less photosynthetic area or less stomatal area to lose water from. You get better spray and light penetration into these, so you get less blonding of the pods, and you can use lower quantities of pesticides. They also have reduced biomass, which makes it easier for mechanical harvest. There are a couple of downsides. Yields may be a little bit lower than normal peas, and sometimes these tendrils will grab a growing point and cause a balling of the plant apex. You'll get a bunch of pods, which can be a problem with mechanical harvest. Now, yield is generally a quantitative trait, but there are a couple of genes that do affect yield, and these are FN and FNA, where the dominant, if you're dominant at both loci, you get 1 flower per node. If you're recessive at either of the loci, you'll get 2 to 3 flowers per node, and if you're recessive at both alleles, you get up to 5 flowers per node. Particularly when you're looking at a processing type where you have a concentrated set, there's only going to be about 4 nodes on a plant which will be harvestable, marketable. And so if you can put more pods at a node, then you can increase the yield. This picture here shows a type with 2 pods per node, kind of this intermediate category of pea here. Disease resistance is another consideration in breeding for peas. There's kind of a basic package that you need for at least what the commercial breeders put in. This would include fusarium wilt resistance, powdery mildew resistance, and then particularly in the West, peonation mosaic virus resistance. Here's a picture of peonation mosaic virus showing the window panes on a symptom on a leaf, and then the other picture here shows a powdery mildew resistant plant in front of a bunch of powdery mildew susceptible types. Depending, some of these diseases are going to be more regional. Bean leaf roll virus is something that's important in the Intermountain West and in places in Europe. Powdery mildew can be regionally important. Pea seedborne mosaic virus is another virus of importance because of its seed-borne nature, as the name indicates. And this can be shipped long distances in seed. When the plants are grown, aphids may move the virus around to other varieties in the field. And so it's definitely a problem if you get it going in your material. So that concludes my talk. I would just point out that there is this review article that we wrote some years back on the origin, history, and genetic improvement of peas, which would have more detail if you're interested in going into more depth with these with pea breeding. So thank you very much for listening.